All right. Hi, everyone. This is part two of our chapter two lecture. I'm picking up where I left off with chapter one. And so it is with this learning outcome 2.5, right? To compare single nucleotide polymorphisms and the various types of gene mutations, including point mutations, insertions, deletions, translocations, in regard to their characteristics and possible clinical consequences. So again, we're not exhaustively, investigating all of these things, but we're going to talk a little bit about some of these different types of mutations and essentially, being able to identify what they are. And possibly, again, like it says here, what types of diseases these types of mutations can lead to. And again, just giving you some examples, right? We're, again, not going to go through every single genetic disease by any means. Okay, so if we talk about mutations, and I think I mentioned the word mutation, in part one, we want to think of, a mutation as being some sort of error in the DNA sequence, right? So it says here a grammatical error in a gene, right? So if we think about DNA sequence as being a sequence of A, T, G, C, right? All those bases that we talked about, right? If instead of, say, there being an A, instead there's a C there, right? Some sort of mutation like that. There also can be a case where maybe one of those bases gets removed, okay? So in any way, shape, or form, these things are going to change that information that the gene is going to convey, okay? And so I kind of gave you some ideas here that they're going to vary in type and severity, and we're going to talk about those, okay? Now, there are repair mechanisms in the cell. And again, that's beyond the scope of our class to talk about, but we could talk about the different types of repair mechanisms that exist. So there are mechanisms that will repair these mutations, right? Or these errors that occur. However, they're not 100%, right? And so there are still mutations that will remain that can't be corrected, okay? And so if these mutations remain and they're not corrected, they could actually be transmitted through generations, right? They're heritable because we're talking about changes in the DNA. Okay, so some different types of mutations. So point mutations is the first one we're going to talk about. And I kind of mentioned this right at the beginning. So this is a change in one nucleotide, meaning instead of there being an A, instead there's a C there, okay? Or there's a G, or there's whatever. So there's just a change in that nucleotide, okay? It says here this will kind of lead to sort of varying levels of severity, okay? Because that single change could actually change the amino acid sequence, right? So, or not, right? So if it doesn't, it's called a silent mutation. If it does change that amino acid sequence, meaning it's going to change the protein that's produced, right? It's called a missense mutation. And you could have benign consequences, right? In the case of a silent mutation, it's not going to affect the protein at all. There's not going to be any clinical consequence at all. However, if it causes a change in amino acid sequence, it could lead to some sort of deficit or not, right? It also still depends, and it could be very severe or it couldn't be nothing at all. So let me just scroll back as a reminder to understand what we're talking about. I realize that this is probably important here. So if we go back just to this picture, I think this is probably all we need. Remember, we have DNA sequence, right? If there's a mutation in this DNA, so we're talking, what this picture is showing you is our DNA in the nucleus. It's showing you transcription, right? Remember, the mRNA is made from the DNA, right? That mRNA then gets translated into a protein. And remember, when we talk about translation, remember, we're essentially reading the information in the mRNA that's in the form of these triplet codons, right? And then that codes for specific amino acids that then make up the protein. So we were just talking about mutations, right? So if there's a mutation here in the DNA, the mRNA is made and that has that mutation. And then when you get here, if say this was supposed to be lysine, right? But instead, when we change, say, this, when this kind of got changed here, it then coded for asparagine instead here, right? Or some other amino acid, it then could completely change the function of the protein or completely make that protein non-functional. Okay, so that's what we're talking about. We're talking about mutations in the DNA, but they will ultimately affect what protein is being made. And again, remember, proteins are the functional molecules of the cell, okay? But however, if you change this information, right, in the DNA, which is essentially the template, and provides that, that information to make the protein, if that gets changed and the protein gets changed, that's what then can lead to disease. Okay. And so that, again, we're just giving an example here of just a point mutation, right? A small mutation. So anyway, keep this in mind when we're talking about the mutations, when we're talking about DNA changes, how that then ultimately affects proteins and then the function of the cell. Okay. Function of cells in general, right? Multiple cells or what have you. So, and then again, remember that these changes in the DNA can then be passed on to generations, right? And so then you could see, when we talk about kind of genetic diseases, you see them in families, right? And you can kind of trace that. So anyway, I hope that sort of helps to kind of put things back into perspective in terms of what we're talking about. So let me go back to where we were. Okay. So I just mentioned this, right? The difference between, and we should know, silent mutation versus a missense mutation, and understand why we call this a silent mutation, right? If there's no change in the amino acid sequence, that means then the protein is not going to be changed at all, and therefore it will function like it should. However, if there's a change in the amino acid sequence, it's possible that that protein would become non-functional. Okay. And then if a protein is not functioning in the cell, that's what then leads to disease. Okay. Here's a term down here, and this is mentioned in the learning outcomes for a SNP, right? SNP stands for single nucleotide polymorphism. And basically this is a point mutation, or it's a change in the sequence, right? But it's present in at least 1% of the population, right? So that you see this change at this certain locus in the DNA. So at this certain place in the DNA, in a certain gene, you're seeing genetic changes that are occurring in more than 1% of the population, okay? And so there's a lot of research studying these SNPs and how they might be related to disease states, okay? So again, if you see this term, that's what that means. And the reason why we call it this and we sort of name it differently here is because, again, it's in at least 1% of the population, suggesting that it may be related to disease. And there are many, we can pull research articles. If you're interested, you could search SNP and probably any disease that you would want, and you probably can find some information about it. I know there's a decent amount of research out there in terms of particular SNPs and Parkinson's, right? And a lot of other neurological disorders as well. Okay. Now so what we just talked about before here were point mutations, right? Just a change in one nucleotide. Now, a different type of mutation, you could actually have nucleotides that get inserted or nucleotides that get removed. Okay. So just instead of just a change from like an A to a T, now you're going to remove a nucleotide that should have been there. Okay. Now these are a lot more serious usually because it will affect the entire reading frame of the triplet codon. So again, if I go back, what the heck do I mean by that? Right? If one of these is removed here, everything then shifts down, right? Because it's always read in triplets. So say you remove the G here, right? Because again, remember, this is removed from the DNA, right? But ultimately you're making mRNA from that. So let's just look here so that we can understand how that might affect the protein. So just looking at this one protein here, if you remove this G, now for this first triplet, it would be UGA, right? Then the next one would be AAG, and so on and so forth. So therefore, it could change all of the amino acids down the line. When we talked about that point mutation, we were suggesting that perhaps just one amino acid might be changed. When you talk about an insertion or a deletion, it could potentially change almost all of the amino acids in the protein. And you probably can then figure out that most likely this is going to lead to the formation of a dysfunctional or non-functional protein, okay? Which then would probably lead to more serious effects on the cell, more serious effects on the tissue, the organ, right? The organ system, and therefore on the person, okay? So that's where we're at with that, okay? I already said this, right? It says wrong amino acids present after where that mutation was, okay? And again, it says clinical outcomes depend on the location in the gene. So like if your insertion or deletion was towards the end of kind of that amino acid sequence for that protein, maybe it wouldn't have much of an effect, okay? But again, you can see how an insertion or a deletion is probably going to have a lot more serious clinical effects than just a point mutation, okay? So again, I hope that makes sense. Again, it's about understanding, right? So if I asked you a question, you know, why would a insertion mutation or why does an insertion or a deletion mutation usually lead to more significant clinical effects compared to a point mutation? We should be able to reason through and answer a question like that, okay? All right. Now, you can also have chromosome level mutations, meaning parts of a chromosome break off, parts of a chromosome break off and then insert somewhere else. So now this isn't just like kind of within a single gene. Now you're talking about like multiple genes, right? Okay. So, and you can actually have a whole duplication of an entire chromosome. Okay. So I'm going to show you a picture in a second that gives you some more examples of that. So here, so now again, shoot, 1 second. Let me fix this. So I know I wrote a label in here, but apparently it for some reason got really small. Yeah, okay. So there you go. You may have to change it in your PowerPoint because I think the one I uploaded had the small words. But anyway, right, types of chromosome level mutations are what we're showing here. So again, this is showing you like a whole part of the chromosome is deleted, right? So this is probably multiple genes here. You can also have a whole part of the chromosome duplicated, okay? You can have a part of the chromosome inverted, a part of the chromosome inserted, okay? So there's all these different types of sort of chromosome level abnormalities that can occur. And again, you might sort of figure out that this may lead to some, have some clinical implications. All right. Okay, so what we just did was give you some examples of different types of mutations, right? Some mutations that are happening sort of within just a single gene on the DNA, and then just a couple of some examples of chromosome level mutations. So again, we should know the difference between a chromosome level mutation versus a a point mutation within the DNA or an insertion just of a nucleotide, right? Kind of nucleotide level. Let's call it that, right? Nucleotide level mutations versus chromosome level mutations. Okay, we can kind of distinguish that way. All right. So the next learning outcome is to now talk about some genetic disorders, right, that are caused by abnormalities of chromosome number. Now, let me just back up for 1 second. There are genetic diseases that are caused by genetic changes on the nucleotide level, right, or single gene abnormalities. Okay, what we're particularly going to talk about here are ones that might be caused by chromosome level abnormalities. Okay, but there are things like if you think about sickle cell, right, or cystic fibrosis. Okay, those are things that are on the sort of nucleotide level or single gene level. Okay, so I just wanted to put that out there. Again, we cannot in one semester talk about every single disease, in a given category. So just like any of the other chapters that we're going to cover, we can't talk about every single disease that exists. We sort of can kind of highlight a few, but and then if you're sort of interested in learning a little bit more about some of these specific diseases, you can do that. And that's why for the final project, everyone's going to choose a disease that we haven't covered. Okay, and then present that so that then we get a little more exposure. All right, but unfortunately, it's impossible to go through every single disease that exists. All right, so I just want to put that out there. Now, again, let's move on. Okay, so if you talk about categories of genetic disorders, I sort of mentioned this, right? You can have these single gene disorders, like I mentioned, sickle cell, cystic fibrosis, things like that. But you can also have genetic disorders that are a result of chromosomal abnormalities, okay? And then you can also have, and a lot of diseases in general, are multifactorial, right? There's some sort of genetic predisposition or some sort of genetic change, but then there's environmental factors that come into play to then actually cause that disease, okay? So that's how you can kind of think about it in these sort of three broad categories. And so I guess I could have added in a little bit of detail here. So like this is something that you probably need to add into your review sheet, right? What's the difference between a chromosomal genetic disorder versus a single gene? right? And what do we mean by multifactorial? These are all concepts that you should understand, okay? And again, it's not about memorizing, me writing a definition here and you memorizing it. We need to understand the differences. Okay, all right, so if we talk about, again, abnormality of chromosome number or structure, okay, it says there is prenatal screening. Yeah, useful for certain disorders like Down syndrome, okay? You know, there's a lot of prenatal genetic screening that's done to detect some of these abnormalities, so that, you know, if it comes up that you have an increased chance, then they'll even do further genetic testing to actually see if indeed, you know, the unborn child might have Down syndrome. Okay. And so you can see here now, we're talking about actual, for Down syndrome, it says it's a trisomy of chromosome 21. There are 3 chromosome 21s instead of 2. Okay. So you basically duplicated an entire chromosome. Other diseases, and again, there's not that many examples of these types of disorders. Why? Because a lot of them end up being embryonic lethal, right? However, so when we talk about Down syndrome, we talk about trisomy of chromosome 21. This is an autosome or a chromosome, a non-sex chromosome. So again, you don't see that many syndromes or disorders that are caused by a duplication of an autosome like Down syndrome, because again, a lot of them cause lethality during the embryonic stage, or you know, soon after birth. Okay, Klinefelter, Turner syndrome, these are examples of diseases that are caused by, you know, chromosome changes in the sex chromosomes, right? So in Klinefelter, okay, you have 2 X's and a Y, okay? So it's a male, but you have 2 X chromosomes instead of just 1, okay? Turner syndrome, right? You don't even have a Y chromosome, so it's just the X chromosome. And these types of diseases or changes in the sex chromosomes are a lot more tolerable. So sometimes you don't even see that in terms of clinical effects. The clinical effects are not that severe, okay? So if you, again, for Klinefelter, the clinical symptoms are pretty subtle. Sometimes though you do have infertility, right? And so sometimes those are kind of more of the issues. In Turner syndrome, okay, short stature, and again, infertility. I can't say that word. But in terms of intelligence and things like that, usually there are no clinical there are no clinical changes in terms of those things. Okay. Now there are other syndromes where you end up getting, you know, 3 X's and a Y, and a lot, and kind of some more changes here. As you start to kind of add some more of those changes, then you start to see an increase in severity of symptoms. Okay. Now for Down syndrome, again, we've probably all seen a patient with Down syndrome, and I don't have a picture here. I guess I could have added it in, but I'm sure most of us are familiar with the sort of quintessential look of a patient with Down syndrome, right? Short neck, they have poor muscle tone, they have wide, short hands and fingers. They also have cognitive impairments. And again, there's going to be a sort of variation in the severity of those cognitive impairments. Most of them also have congenital heart abnormalities that usually require surgeries, hearing and vision problems, and there are other symptoms and signs as well. Okay. So there is a picture in your text, figure 2.15, that shows you a picture of a young girl with Down syndrome. And again, so if you've never really, or you're not really sure what I mean in terms of what a patient with Down syndrome, kind of those characteristic features, search it and look up a picture. But probably most of us have seen a patient with Down syndrome. Okay. All right. So to move on, again, an example there. Okay, so I already mentioned this, right? I mentioned this sort of single gene mutations. Oh, I guess we are going to talk about those a little bit more. I thought for some reason it was sort of, I didn't talk about it that much. But so the single gene mutation examples, again, things like cystic fibrosis, right? Sickle cell, okay? You'll see that the pattern of inheritance will vary, okay? And so these are some examples here. We talk about them as being autosomal recessive or autosomal dominant. Again, remember, autosome right, means it's a non-sex chromosome, okay? If you talk about X-linked here, X-linked recessive, this means that the gene is on the X chromosome. Here, the gene that's affected is on the autosomes, right? So I'll show you, when I show you a karyotype, did we look at a karyotype? Okay, let me just show you this right now. I know I have it in here, right? So again, so you know what the heck I'm talking about. So and we'll talk, I'll flip back and we'll talk more about the karyotype when I get to it. But for karyotype, right, what this is showing you is this is showing you the chromosomes, right? Remember, we have 23 pairs of chromosomes. There are 22 autosomes, right? So you can see all the autosomes here. And then these are the sex chromosomes, right? So this is showing you in a male, right? An X and a Y. If it was a female, you would have 2 X's here. Then this is showing you an example of a karyotype with a patient with triple X syndrome. Again, this is another type of syndrome that's associated with the sex chromosomes. Okay, so anyway, I'll talk a little bit more about that, but I just wanted to show you because I realized it makes sense to show you that when I'm talking about autosomes versus sex chromosomes. Okay. All right, so that's what we mean by autosome. You can also have diseases that are linked to genes that are found in the mitochondria or in the mitochondrial DNA as well. And remember that those are always passed on maternally, okay? Some other terms down here, again, like I told you, I know this is a lot of information and very sort of basic genetics heavy. And I realized kind of as I was going through that maybe these terms weren't even really mentioned. If we talk about genotype versus phenotype, right? Genotype, we mean what's in the DNA, right? Or what alleles do you have? Phenotype is talking about what's expressed, right? Do you have the disease or don't you? If we want to think about that within the realm of pathophysiology. Remember, if you talk about homozygous versus heterozygous, we can talk about homozygous recessive, and I'll probably have to go through this and pull up the whiteboard during class. I probably will. But say we're thinking of, let's just take my cystic fibrosis example, and we can use C's, right? And if we say that that's an autosomal recessive disorder, okay, we can call the recessive version, right? You know what? Let me just do this right now. Let me just add a text box. Let me make this bigger. Okay, so if we talk about, I'm just going to do this because I think this is the easiest way to go about it right now. Okay, so we're talking about, so say we a given genotype, right? Again, we're going to think about the gene for cystic fibrosis. Okay, the normal gene is a capital C. Okay, the lowercase c, the recessive allele. So again, remember you have alleles, which are alternate versions of a gene, right? So if we're talking about inheriting one copy from our mother and one copy from our father, okay? So one of these is from your mother, one is from your father, right? This is the dominant allele here, the big C, and then the little c being the recessive allele, okay? So if we looked at this genotype here, we would say this is heterozygous, okay? You have one dominant allele, you have one recessive allele. If we say we have 2 big C's, right, as our genotype, so we got one big C from our mother, one big C from our father. Remember, these are considered alleles. They are alternate versions of the gene. And again, for our purposes, we're going to talk about the normal allele versus the diseased allele, okay? And so in this case, you would be considered homozygous dominant, okay? And so now, again, we're thinking of something that's an autosomal recessive, my cystic fibrosis example. Now, if we talk about 2 little c's here, this is the genotype, again. So now you got the diseased allele from your mother, diseased allele from your father, okay? And so we would call this homozygous recessive. Now, in this case, for an autosomal recessive disease, okay, which genotype would have the disease? Okay. If you're homozygous recessive, you have the disease. Okay. If you are heterozygous, right, big C, little c, you are called a carrier, right? You don't have the disease, but you can pass that little c on to your offspring. If you are homozygous dominant, right, you do not have the disease. Okay, so again, when we're thinking about pathophysiology and we're thinking about genetic diseases, we talk about autosomal recessive. There are other types of diseases that are inherited this way. Okay, so that means the only people that have the disease are those with this genotype. Okay, they have 2 recessive alleles, 2 disease alleles. Now, if you talk about autosomal dominant, it's different. This genotype, if you have just one copy of that dominant allele, which is the disease allele in this case, you have the disease. So for autosomal dominant disease, big C, little c has the disease, they're affected, and big C has the disease, they're affected. The only ones that are not affected are those that have the genotype of those 2 recessive alleles. Okay? In the case of X-linked, remember that's just, it's on the X chromosome. Okay, so let's move on a little bit more from that. So again, if you have more questions about that, we can go over that again, but hopefully that helped. Let me move on now. Okay, so we just talked about this, autosomal recessive, right? The affected gene is present on a non-sex chromosome. That's what we mean by autosome. It can affect males or females, doesn't really matter. Again, recessive inheritance, I told you that, okay? It says trait expression requires defect of both gene copies, right? That means in order to have, if you have the disease, if you have both of these recessive alleles or the disease alleles, okay? That's what that means. If an individual inherits only one copy of the mutated gene, meaning you're heterozygous, right? Big C, little c, you're a carrier. You will not express the trait. You will not have the disease, like I said before. If both parents are carriers of a gene mutation, right? So you have 2 heterozygous parents, big C, little c, right? Both are heterozygous, big C, little c. They have a 25% chance of passing that on to their offspring, or the offspring have a 25% chance of having the disease. Okay, again, you can do a Punnett square to figure that out. Perhaps maybe that's a little bit beyond the scope, but something like that and understanding this on that level is probably helpful. And so we can do that in class, and perhaps I probably will bring this up and maybe have you guys do a Punnett square. Okay, all right. So one of the ways of sort of looking at or tracking genetic disease in families is kind of making up a pedigree here, right? And so if you went to a genetic counselor, you know, this might be something that they would do. And again, it's important for you guys to understand these concepts, because if a patient has a, you know, you suspect they have a genetic disorder or they were diagnosed with one, it's important that they actually do see a genetic counselor, okay? So if you look at an autosomal recessive pedigree, this is kind of what's showing here. So a few things to understand. Square is a male, circle is a female. Empty means unaffected, does not have the disease. A dark shaded one means they have the disease. And if you're looking here, it's showing you 2 little F's. I gave the example of showing you 2 little C's, okay? So this is just showing you that these are the only 2 that they actually know the genotype of them because they have the disease, right? It's 2 little F's here. Okay, so that's what this is showing you here. Again, these are all unaffected in terms of the white circles, the dark being affected. Okay, if we talk about autosomal dominant, I already went through that with you. So it can affect males or females, dominant inheritance. You only need one copy of that mutated gene or that diseased allele to have the disease, right? Remember my example. If you're heterozygous with a big C, little c, you have the disease. If you're homozygous dominant with 2 big C's, you also have the disease, okay? It says here, if one parent has an autosomal dominant gene mutation, okay, just one of those big C's, 50% chance of offspring having that disease. Okay, so some examples, Huntington's disease, Marfan syndrome are some examples here of autosomal dominant. There are other examples of autosomal dominant diseases as well. And what you can notice here from this pedigree, again, same means the same thing, right? You got shaded meaning affected, you have the disease. Non-shaded, no disease, female, male, right? Female circle, male squares. You can see that there are a lot more affected individuals within this family because this is an autosomal dominant pedigree, okay? All right, if we talk about X-linked, I mentioned again this in the beginning too, or sex-linked, the mutations will occur on the X chromosome, okay? It says traced through the maternal lineage, Y, X chromosome, right? Females have two X chromosomes, right? Males have an X and a Y. So because of that, you primarily see effects in males, okay? So it says here, female carriers may manifest clinical symptoms, okay? So you could have one normal X allele, right? One normal sort of allele and then one disease allele if you're a female because you have two X chromosomes, and maybe you express some symptoms of the disease. If you talk about males, they only have one X, right? And then one Y. So they only have one chance of having that normal allele. So if they have the disease allele, they don't have another X chromosome to basically sort of compensate for that. Okay, so an example, Fragile X syndrome. There are other examples as well, like color blindness. Okay, there are many other examples of sort of X-linked disorders. Again, we're not listing them all. I don't expect you guys to memorize them all. That's not what it's about. It's just about understanding if you have a patient that has a certain disease and then you know it's X-linked, you should know what that means, okay? You should know that you're going to see it more in males and probably not in females, okay? And why that's the case. I mentioned this also too, that you can have disorders that are traced back to the mitochondrial DNA, okay? And again, these would be inherited exclusively from the mother, okay? If you have an affected male, it won't pass on that male will not pass on the condition again because it's part of that mitochondrial DNA. However, affected female may pass that condition on in about 100% of the cases, okay? It says it may be inherited in an autosomal pattern, okay, because it's not really X-linked, but it's kind of has this sort of very it's unique, right? Because again, the DNA is coming from the mitochondria, and again, it's only going to come from the mother because again, you have those mitochondria in the egg cell. Okay, we're not going to really talk specifically about diseases that are associated with the mitochondrial DNA, again, beyond the scope of our course, but something to, again, be aware of and know about. Okay, if we talk about multifactorial, right? You have genetic and environmental components. So most diseases are multifactorial, okay? It says you still may see disease patterns in families. That's true, right? Because you have some shared genetic factors and probably shared environmental factors as well. It says here, lifestyle and environmental modifications may be actually more useful than clinical genetic testing alone because there is no one gene to pinpoint this disease, okay? Again, it is not like Down syndrome where trisomy of chromosome 21 caused Down syndrome. These are diseases which are listed here. Things like hypertension, inflammatory bowel disease, diabetes, even cancers can be listed here, where there is no one gene that's going to cause this, okay? So more important to live a healthy lifestyle and basically alter those environmental factors that could come into play, because there's nothing you can really do genetically, okay? All right. So this next learning outcome, differentiate the mechanisms responsible for phenotypic variations in human disease. So again, if we think about any sort of, most, probably all human diseases, there's always a variation, okay, in sort of disease severity. Okay, that's just normal. Okay, and so so we can talk a little bit about that as to why, okay? I mean, there's not really any clear-cut, I guess, reason why, okay? Again, and I think we kind of just said this. So variation in multifactorial disease presentation, genes, environmental influences, lifestyle choices, those are reasons why we see such a variety in disease presentation, okay? Even in single gene disorders like we talked about, okay, you still will see variation, okay? So again, there's still some environmental influences influence there. There's also something called reduced penetrance. And so what this means is that some people with the mutation have the disease and others don't. Even in these sort of single gene disorders that we really can pinpoint back to a single gene, you still can see variation. Okay, so again, there's still always the influence of environment. If you talk about cancer syndromes that have sort of been linked to some more specific genes, right, like the BRCA genes for breast cancer, you again can see that a patient with a mutation, right? Patient has a family history of breast cancer, goes and gets genetically tested, has a mutation in one of the BRCA genes, they may not ever develop breast cancer, okay? Or that type of breast cancer. And there are different types of breast cancers, but particularly the one that's been associated with these BRCA genes. All right, again, there's going to be differences. If you talk about the term called variable expressivity, the severity of the disease will vary, right? So for muscular dystrophies, Duchenne versus Becker, they're still going to affect the dystrophin protein. That's where the name comes from. But depending on, you're going to have a variation in the severity depending on that mutation and kind of how affected that protein is. Okay. And it says here, phenotype variation may also be unexplained. Like I said, kind of in the beginning, we know there's phenotypic variation. That's just a known thing. A lot of times we don't really know exactly why. Okay, so I kind of just gave you some examples of this here. Okay, the next learning outcome, understand the role of genetic and epigenomic factors in cancer. Okay, so I kind of just alluded to that, right? I mentioned that those BRCA genes, okay, and it sort of comes back here again. It says here, it's cancers caused by disruption of normal gene function. Yeah, and usually in genes that function to control cell growth, okay, and differentiation, okay? And that will vary in terms of how that gene function or that cellular function is disrupted, right? Like I said, the most sort of common mechanism is mutations in genes that will control cell growth or control cell proliferation. Okay, and there are kind of two big categories that you talk about when you talk about these genes that control cell growth. Proto-oncogenes are genes, these are normal genes that everyone has in their genome that are necessary to control cell division, okay? Proto-oncogenes will normally increase cell division, right? So they're going to cause more cells to be produced. And there are many times in your life, in your body's sort of lifespan, that you want to increase cell division, okay? So these are normal genes. There are also tumor suppressor genes, which normally will just decrease cell division because there are times where you need to slow down cell division. Okay, what happens with cancer is you have mutations in these genes that then alter the function of them and can lead to overproduction of cells, right? Or increased cell growth that's totally uncontrolled. Okay, there is clinical genetic testing that's especially in the last probably 10 years that has pinpointed certain genes to certain cancers. Okay, so I mentioned this already. For breast cancer, there is a certain specific type of breast cancer that has been connected to or related to these BRCA genes. Okay, and so again, if you have a family history of breast cancer, you can go and get tested for a mutation in these genes to know if you're more susceptible. Again, that doesn't mean that you're going to then develop the breast cancer. It just puts you at an increased risk. Okay. Now this gives you some examples of some different tests. Okay, so clinical tests, sort of genetic clinical tests. Again, some just basic examples, doesn't really go into much detail. This is again something that you could kind of go into more detail on your own, but again, be sort of beyond the scope. We're meant to kind of just touch on this and give you some examples. And the idea is to be aware that there are, genetic testing options for patients, and that if you suspect some sort of a disease that is genetic in origin, that patient should then be referred to a genetic counselor who will go over all of this stuff with them and do some of these tests, okay? So some examples, right? Karyotyping, I sort of just quickly showed you a karyotype before. I'll talk about it again. Fluorescence in situ hybridization, or FISH. Also single gene testing. And then there's also some sort of genome-wide association studies that have been done. Okay, so there is a field of study that basically kind of, it's called cytogenetics, right? So we're essentially studying the the genetics inside the cell. Okay, so this sort of cytogenetics field is the field that develops these types of genetic testing tools, okay, and also sort of performs them on patients and analyzes them. Okay, so maybe that that explains it a little bit better, right? So if you want to examine chromosome structure and number, you can use karyotyping, which is just a visual examination of the chromosomes, right? It'll show you, again, basic structure of those chromosomes and the number of the chromosomes, okay? So you can identify gross abnormalities, right? Like trisomy of chromosome 21, or even, it's mentioning here some terms that didn't come up before, triploidy, aneuploidy, meaning triploidy just means that you have 3 chromosomes instead of 2. If you talk about aneuploidy, it just means there's an abnormal number of chromosomes, okay? So basically you can figure out how many chromosomes you have and if there's an abnormal number at any given chromosome, okay? You cannot detect single gene mutations with a karyotype, okay? So again, these are for chromosomal abnormalities. So I showed you this already. Here, again, we can look at chromosome number. So we know we have 22 autosomes. We should have 2 copies of each chromosome. You could also see gross abnormalities, meaning like if something was broken off or something like that, you're missing a part of a chromosome, you could see that. But if it's something subtle, a karyotype is not going to show you that, okay? Then remember, again, these are the autosomes, these 22, and then the sex chromosomes, okay? So again, if you have an X and a Y, a male. What this is showing you is an abnormal karyotype. You should be able to recognize an abnormal karyotype if I showed you. And this is actually particularly showing you a patient with triple X syndrome. So there are some other examples in the book. This is just one that I pulled, okay? If you talk about FISH, which I mentioned, this also is a way of examining chromosome number. It's just a more specific way, and it's not like a visual way. It's more of sort of an in vitro kind of in the test tube way of detecting abnormalities, okay? And it's much faster than the karyotype. So it says here, you can find those abnormalities with FISH that you couldn't see with a karyotype. And like I said, it's also quicker, okay? So you can detect trisomy 21 Down syndrome with using FISH rather than doing a whole karyotype, okay? So there are these fluorescent probes that are used to identify the chromosomes and specific genes that you then can analyze. Again, technique beyond the scope. Exactly how the technique works is beyond the scope of our class, but know that this is a way of identifying things like Down syndrome, okay? But again, and looking at other, maybe more subtle abnormalities that you can't see with a karyotype. But again, this is really, again, for chromosomal abnormalities, okay? Like I said, there are also techniques or technologies that can do single gene sequencing. And so basically you can look for nucleotide changes anywhere in the gene, okay? It won't detect deletions or rearrangements. There are lots of techniques that are used to do sequencing, and there are new technologies that really are always being developed. So you can kind of, you can sort of use, you know what, I'm not even gonna go through the technologies, don't worry about it, but it is sort of ever changing, and we have a lot, again, if you're gonna compare our sort of abilities 10 or 20 years ago versus now, we really have the technology now to detect some of these single gene changes where we didn't, say, 20 years ago. And again, I'm sure in another 10 years, we're going to have an even newer technology that's going to be even more useful. Okay. Now, this learning outcome is going to now talk about therapies. Okay. So what we mentioned here was, did I miss one second. All right, sorry. I thought I had added something in about if you go back here, right? We mentioned here genome-wide association studies and sort of genomics, okay? There it talks a little bit about this HapMap project where they really are focusing on that. Remember that 0.1% of the DNA that's different between individuals and really studying that on a sort of genomic level. So the genome-wide association studies and this sort of HapMap project, if you look in your book on page 41, is more large scale. Okay. And you know what? It's okay that I probably didn't go over it in detail. We don't need to know everything about it, but maybe it's towards the end of maybe on the last slide. All right. So, sorry, I just want to make sure I mentioned it if it's not on that last slide before I forgot. Okay. In terms of now therapies, right? So it says apply the following genetic based, sorry, I have a tickle. Okay. Sorry, I had a tickle in my throat. So, in terms of trying to apply some of these therapies to appropriate clinical situations, again, are we going through like every clinical situation and how we might apply it? No. But if we consider everything we sort of learned in this chapter, and we are concerned that a patient might have a genetic disease, again, important to then refer them to a genetic counselor or some other appropriate medical professional, maybe an MD that is specifically specialized in genetic diseases. All right, so some of the things we'll mention here: gene replacement therapy, pharmacogenomics, right? Antisense nucleotides and transcription factor modulation. Okay, so these are just some examples of some genetic-based therapies, and I think this is an ever sort of evolving field. And just like we said with the technology about sequencing, these technologies are going to continue to get better as well. Okay, and we're going to come up with probably more and more therapies that are genetically based. Okay, if you think about gene replacement therapy, this is the idea of expressing, right, or silencing disease-causing genes. Okay, so when you're saying expressing, you would want to express the normal allele, right? Silence the diseased allele, okay? And you can potentially do this by actually introducing genetically engineered DNA or RNA, and basically inserting this into a virus, a sort of a virus that's not going to cause disease, that then can get in or nearby those cells, and other cells the virus can then infect, right, cells, the host cell, and then inject that DNA into the host cell, and then potentially that host cell can incorporate that engineered DNA, which has the normal, allele in it, and then that could potentially rectify the function of those cells. Okay, there are a lot of limitations with this in terms of the success of that DNA integration. A lot of times it really doesn't get integrated into enough cells to have a clinical effect. Potentially there is harm from the virus, right? That virus could go through some sort of, change, what have you. I think the biggest thing though is really the limited success of that DNA integrating in. But this is kind of one technology that is sort of being investigated. Okay, as of now, it probably hasn't had as much success as we hoped. Okay, if we talk about pharmacogenomics, right? Personalized medicine. We've probably heard a lot about this over the last couple of years. And it's the idea that it's blending pharmacology, right? Basically, drug development with genetic or genomic capabilities. Okay. So some of the recent focus here, finding genetic variants in a drug metabolic pathway. So there are alterations in the way a patient might clear a drug, for instance, right? And the way the drug gets metabolized in the body. And so important to identify these sort of genetic variants. And then if you know that patient has that genetic variation and they're not going to metabolize the drug in the way that we might traditionally think, then maybe you then offer them a different drug, okay? Or something to try to sort of deal with that change in metabolism, okay? So that kind of goes along with this, right? Predicting medication response. So again, knowing the patient beforehand, knowing that they might have some altered response to a medication, again, either you're going to prescribe something else or you're going to offer some other therapy along with it to try to negate that sort of alternate or aberrant response. So that's, again, this is kind of going along the same thing, right? Customizing prescriptions based on genotype, particularly things like anesthesia. I know when this genetic counselor came and spoke to us for our Women in STEM panel, I don't think last year, but the year before, she was saying that there are very easily can you do a genetic test to determine if you have this mutation that will alter your response to a particular type of anesthesia. And it could potentially be a life-threatening response, right? And you would never know this because how often do people go through surgeries? So the only time you would know it is after you've given that anesthesia and then potentially might have some life-threatening response. So if beforehand you knew about it, well, then the doctor's not going to use that type of anesthesia. So kind of same thing to go along with chemotherapy. There may be some types of chemotherapy that are more effective for some individuals versus others. So again, if we know that ahead of time, we then can design those prescriptions or that therapy to that person. Okay, so that's really what personalized medicine means. Okay, there's some other examples here. Antisense oligonucleotides. So it says they're used a lot in cancer treatments. And basically, it takes a sequence of complementary nucleotides, meaning if we're talking about a sequence, say, in DNA, ATGC, it's going to take the complementary sequence, right? I forget what I now said, TACG, right? That complementary sequence. And what it can do is that sequence can sort of bind to that, the target, right? Via complementary base pairing, and it can actually block the gene product that's the disease gene product or the aberrant gene product from being produced. Okay, so again, you can see another approach that uses genetics essentially, right? Because we're talking about kind of DNA and RNA level. Also, transcription factor modulation. So this is saying it alters specific gene transcription levels. So transcription factors are essentially proteins within the cell that will bind to the DNA. And they'll bind to the DNA and they will then either increase the transcription of a gene, right? And therefore the production of that protein, or they might decrease it. And so apparently there are lots of, I guess this idea is that you're sort of targeting or using or designing drugs that act like transcription factors or interact with transcription factors particularly. And again, why do we talk about that as being sort of genetic based? Because transcription factors are always going to interact or associate with the DNA. Okay, it says here they're not cell specific, and because they're not cell specific, there is a great potential for side effects. So it gives an example in the book of prednisone. Right, prednisone is a steroid. Okay, and so steroid hormones will actually enter into their receptors, actually sit inside the cell, and they'll enter into the cell, bind to those receptors, and a lot of times once the prednisone binds to its receptor, it'll actually go to the nucleus and act as a transcription factor. Okay, so that's why it's giving you this example sort of related to transcription factors. Has anti-inflammatory effects, right? But it also can lead to weight loss and bone loss, again, because it's not cell specific. Okay, so you can kind of see how there's limitations to every single one of these technologies or these therapies that we mentioned. Okay. Oh, all right. So I think this is where it mentions this HapMap and the genome-wide association studies a little bit. Okay. This is the last part of the lecture. So it says analyze the impact of advances in genomics, right? Remember, this is large scale and epigenomics on personalized healthcare. Yeah, sorry. I knew I had a slide of it, and so that just made me nervous that that for some reason I didn't have it. So it says here, since the completion of the Genome Project, okay, more than 1,800 disease-associated genes have been identified, right? And around 2,000 diagnostic genetic tests are available because we know the entire genome. So before we had that human genome sequence, we weren't able to identify all these disease-associated genes and then identify or why can't I even think of the word? Anyway, we weren't able to identify these diagnostic tests, right? Or come up with or design or invent these diagnostic genetic tests that are around, okay? I already mentioned this before, but humans are 99.9% identical. This, and again, I mentioned this when I thought I didn't have this slide. There is this international HapMap project that was launched. And again, its focus is to analyze the 0.1% of genes that make us different from one another, right? And really kind of look at this on a larger scale, okay? So in terms of like why we would want to study sort of genome level changes, right? Because we can sort of detect general risk factors, right? You might be able to diagnose rare and common disorders by just understanding, how disease, the sort of larger scale of disease and sort of genetic factors, okay? Some of the data from this HapMap project revealed 3.1 million common SNPs. Remember I mentioned SNPs, single nucleotide polymorphisms in the human genome, okay? Oops, I got a typo here. In geographically diverse populations. So again, because they're focusing on this 0.1% of genes that make us different, they actually were able to identify these genetic changes that then can be linked to disease, okay? Remember, the SNPs are changes that are present in more than 1% of the population. Okay, I think finally this is the last slide. If we talk about genome-wide association studies, again, these are particularly going to focus on those SNPs that have been identified and then associate these SNPs with specific traits, okay? I guess you can kind of think of the HapMap project as sort of initially identifying these SNPs, and then these genome-wide association studies as now associating these SNPs with specific traits or specific diseases, okay? So again, what are some of the implications? Useful for identifying causal genetic factors, meaning if you have this, single nucleotide polymorphism in this one specific gene, it may, again, predispose you for a certain type of cancer, okay? Also, it says identify common genetic variants in disease. So if we're talking about a disease population, you may see that those with the more severe form of the disease have this one sort of polymorphism or this one nucleotide at this one spot, and those with less severe cases might have a different version, okay? And again, you can kind of use this for studying both rare and more common disorders. All right, so I know that was a lot of information in one single chapter. So there probably are some questions. Please ask them. Let me just go back one more place here and just show you one more thing. I think I mentioned, yeah. So let me just add. Yeah, I'll probably have to do that in class. I want you to know that in your textbook, and I will do this in class, the Punnett square, I'll just make sure we all know how to do that. But when this is mentioning here, right, 25% chance of disease manifestation in the offspring, you can illustrate this doing a Punnett square, okay? In your book on page 36, it gives you an example of that. I think the reason why I didn't put it in the lecture is because it's better to do that sort of live. But I may actually insert just an example of it, but you should totally understand that, okay? So, well, you know what? Let me just try to, I probably can just do it quickly. I just feel like if I don't put it in here, we go with our example, right, that we had before, okay? So remember, it's, right, our big C, little c example, okay? So there it is. So, and again, this is going to be kind of a little bit of a crappy Punnett square, but we'll draw it out in class as well. But if we talk about now, again, this is paternal, this is maternal. We're talking about, again, different alleles, right? One dominant, one recessive. And then you want to look at now the offspring. What are the possible combinations that the offspring could have, right? So the possible combinations are big C, Okay, if we go over here, and we can space this out a little bit, and we can space this out, okay? If we go over here, we're going to take this little c and this big C here. So this would be big C, little c, okay? Now if we go down here, oops, I don't know why I did that. We don't want it to do that. Okay, this now will be big C, little c. And then over here, okay, you're going to have little c, okay? So these are the 4 possible genotypes that could be produced from parents that are carriers, okay? And so again, remember what we talked about when we talked about the autosomal recessive diseases, okay? Anyone that has the dominant allele, the big C, is not going to have the disease. We call heterozygotes carriers, right? And that's what we placed up here, 2 carriers. What's the possibility that their offspring, or what's the percent chance that their offspring will have the disease? Well, if you look at here, right, the only genotype that will have the disease is this one here, right? So in this case, right, it's 1 out of 4 or 25%. So that's where that comes from. So this is a Punnett square. It's just lining up the 2 parents and then looking at the possible genotypes that could be formed. Then you have to look at those genotypes and think about phenotype, right? Will they have the disease or not? All right. So again, big C, that is homozygous dominant, no, does not have the disease. Big C, little c, heterozygous, carrier, does not have the disease. But however, if you're homozygous recessive, you have 2 little c or diseased alleles, you have the disease. Okay, so again, 25% chance because it's 1 out of 4. All right, so we can, I guess what I'll do is I'll just, oops, see now I screwed that up. I just wanted to write in here that this is our Punnett square example. Okay, that's what this is. All right, guys, so I will end